Good afternoon, everyone. My name is Anita Ramani, and we're here to chat about the genetic components of obesity. The objectives that will be covered today are describe and discuss the impacts of obesity on health and mortality, explain the difference between a gene, a variant, and a mutation, communicate the difference between the FTO variants, and describe the impact of the FTO gene mutations on our health. Before we dive into the lecture today, I wanted to cover a few terms that you will hear me bring up. The first one is body mass index. Body mass index is a common tool that we use in healthcare to assess the risk of an individual when it comes to their weight and their risk of disease. So for example, if their BMI is under 25, the risk of disease is much lower than, say, an individual who has a BMI of 35 or greater. So we assess the BMI by simply taking an individual's height and weight. And you may be wondering, what is my BMI? Well, at the end of the lecture, I do have a link to a calculator that you can assess your own BMI. So if your BMI is greater than 25, you are considered overweight. If your BMI is over 30, you are considered obese. So looking at this chart, the prevalence of obesity in males and females according to their age and geographical region, this is a great chart that has a lot of information on it, showing us the trends that we have been experiencing from 1975 to 2015. So for men and women, this is showing us data with BMI greater than 30, so again, obesity. So the population has experienced a steady increase in weight over the last 4 decades, and it has affected adults, children, adolescents. No one has been spared. And then on top of it, shown on this graph, it's giving us a worldwide view. As you can see at the bottom, South Asia, East and Southeast Asia, Latin America, the Caribbean, high-income Asia Pacific, high-income Western countries. They've included multiple populations in showing us this information. Unfortunately, at this rate, if we continue in 2025, which is literally around the corner, 1 billion adults, which is 20% of the world's population, will be obese. As a result of this, we are seeing more and more health complications, including type 2 diabetes, heart disease, multiple cancers, COVID complications, and premature death. Now, COVID complications, that's definitely a newer discovery since the pandemic. The connection is that the more obese an individual is, the more complications they have, and they have a higher risk of hospitalization on top of it. And also there are secondary complications such as osteoarthritis and more difficulty getting around, or for example, sleep apnea is another concern. Now, the interesting thing is that over the last few years, it has been discovered that in addition to an environmental component, there's also a genetic component to obesity that we did not know about before. Genome-wide association studies help scientists identify genes associated with particular diseases. So what scientists do in a really quick and easy way, it's obviously more complicated than this, but they're able to view the DNA as a whole, and they review populations to look for different variations in the gene. So through these studies, disease-causing genes are identified. So I've thrown a lot at you, so let's just backtrack a bit. So what is a gene? A gene is a unit of heredity. What is a variant? A variant is a gene that may have one or more mutations. Now, what is a mutation? A mutation is a permanent alteration to the DNA sequence. So basically, scientists, when they look at DNA across multiple populations, they are looking for a permanent alteration to the DNA sequence. So they found that over the years, they found through the genome-wide association studies that there are more than 1,100 loci associated with obesity traits. What does this mean? That means that there are 1,100 specific genes that are in specific positions on the chromosome that have an impact on obesity. And as you can see from this graph, that, for example, the purple there is a correct copy of a gene, but the yellow shows that the mutation or the permanent alteration is in its place. I'd like to take us back to when the first obesity gene was discovered. It's an interesting story. It was first identified in a study of a fused toe mouse. So you'll hear FT for fused toe. So this gene that they found received an FT label. So this mutation that they found was inserted into a gene, which led to a deletion of another gene. So when this gene was first cloned or copied in 1989, they originally thought this particular gene had a role in cell death. But eventually, through this process, they found several genes at one time. So on this chart here, there are 4 genes that were discovered. So the original name for the first one was FT1, and then they changed it to FTS. And the second one was named Phantom, so received the name of FTM, the abbreviation. Eventually, it was renamed to RP-GRIP-L1. The third one, that's another interesting story. So at first, when this gene was seen, it was the largest of all of the genes, so they named it FATSO. And they gave the abbreviation of FTO. And then I'll come back to what it was renamed to. And then the last group was called the IRXB cluster. And those are the 3 that you see that it was just, named individually, but they still do call it the IRXB cluster. Now, they started discovering that there was something to do with obesity, but they weren't sure. So before the first report about this association between the gene and its possible connection to obesity was published, Andrew Hattersley, one of the authors of one of these papers, realized the problem in keeping the name FTO, or the gene, with its possible connection to obesity. So in the end, he renamed it in a clever way, based to keep the old symbol of FTO, but to change the name from FATSO to fat mass and obesity related. Now that we've learned a little bit about the history of these genes, we can take a look on the chromosome to see where these genes are located. So you see FTO is in the center there, and downstream from there you see the clustered genes IRX3, 5, and 6. And then further up from FTO you see the RPGRIPL1 gene. So the theory is that the FTO gene affects the cluster genes as well as the RPGRIPL1 gene, but the mechanisms are not exactly for sure what it does, but we do know it has an impact on obesity. So there are studies that have been done where, the gene has been removed and, a deficiency occurs and this is what happens. So we're like, okay, so these genes impact obesity. And if we remove them, then positive results occur. Well, not always. So they created a mutation, a genetic deficiency of IRX3. Now, a study did show 30% weight loss, but when the study was replicated, contradictory results. So they haven't been able to consistently show that creating this deficiency allowed for the weight loss. Now, they did, when they altered the RPGRIP1L gene, they did find that individuals did have more weight gain and uncontrolled eating. I am now going to head into a bit of a discussion about ancestry and various populations, and when it comes to the FTO gene and its impact on BMI. And if you remember, that means body mass index. So there have been multiple studies over the years to explore FTO's role with obesity risk across populations. Genetic studies have shown that FTO is an important gene when it comes to obesity risk. However, the issue is trying to replicate those studies is where this issue lies to get consistency. So through replication studies, it has shown researchers that the FTO gene does have a consistent effect when it comes to obesity, but for whatever reason, when it comes to African Americans and Africans, the connection is not as consistent. But as we learn more about the impact of these genes on obesity, we will be able to better understand the connection. So moving on to the next slide, I would like to end this talk with two studies that were led by UCL scientists, the Medical Research Council, and the King's College London Institute of Psychiatry. These studies show science in action. "359 healthy male volunteers were chosen for this study. They studied two groups of participants. those with 2 copies of a high obesity risk FTO variant and those with a low obesity risk version. So the group with the high obesity risk FTO variant is known as the AA group here. And you'll see this on the graph in front of you. So there's the AA group and the T group. And the AA group is the one with the variant. So the first study involved a group of 20 individuals, so 10 and 10 for each group, and they fasted overnight. They were asked to rate their hunger before and after the meal. So every 20 to 30 minutes, their hunger was evaluated and blood samples were taken of a hormone called ghrelin. Now you may ask, what is ghrelin? Well, ghrelin is a hormone that's released by your stomach when, for example, your blood sugars are low, where you're physically hungry. Ghrelin is released to say it's time to eat. And how I tell my students is, if your stomach's growling, that means ghrelin has been secreted, the G, growling ghrelin. So I usually get a laugh out of that one. And they remember it for their quiz or test. So success. So we noticed, in the study, they noticed that meal ingestion suppressed ghrelin levels in the TT subjects. So when they fasted overnight and in the morning they were hungry, so their ghrelin levels are naturally high. So when they eat, you expect the ghrelin levels to go down. Well, in the TT group, that's exactly what happened, but for the same number of calories, the AA subjects' ghrelin levels continued to be high, and they felt hungrier after the meal. So these findings suggest that altered circulating ghrelin post-meals might contribute to altered appetite and food intake that we see in AA subjects. So basically, they're eating more because their ghrelin levels continue to be high. So the results of the study suggest that obesity risk variant group, which is the AA group, does not suppress ghrelin in the normal way after a meal, which leads to a change in appetite and food intake, which would be higher because their ghrelin levels are higher. And if you look at this chart in front of you, it shows, so the red is the AA group and the TT group is the blue. And so you'll notice that their hunger is higher, their ghrelin levels are higher, and as the minutes progress, they still continue to be higher than the TT group. And then you notice the D and G. It's also, the ghrelin level is suppressed with the TT group, but continues to not be as well suppressed in the AA group. So even without this gene knowledge, do you see yourself in the AA or TT group when it comes to food intake? After you eat a meal, do you feel satisfied or do you still feel hungry after you eat a meal? And you could even do the comparison with you sitting next to a sibling or a spouse, and if you consume the same amount of food, do you notice, or does your spouse or sibling or friend notice, are they just as satisfied? So it makes you think there that even without having the genetic variant present, or the knowledge of the genetic variant present, do you fit more in the TT or the AA group? So this is also, again, the next study. So then in a different study group, functional MRIs were used with 24 individuals to measure how the brain responds to high and low calorie food images, as well as non-food items before and after meals. So participants were asked to rate the images on how appealing the images were, and then blood samples were again taken. So individuals with the obesity risk FTO variant, this again is our AA group, rated pictures of high-calorie foods as more appealing even after a meal than the low-risk group. So you can see their brain responses in real time. Their functional MRI results reveal that the brains of the two groups responded differently to the food images too. So the differences were most noted in the brain's reward centers. These reward centers respond to alcohol and recreational drugs in the hypothalamus. And so if you look here, that even after both groups consumed food, and the TT groups, their ghrelin levels dropped, and the AA group, their ghrelin levels were still higher. So when they saw images of high-calorie foods, their brains were more excited because they were still hungry, per se. So this was an interesting, graph which shows the excitement there. And then going back to this again, so you see here with the AA group that they have this appeal for high-calorie images compared to the TT group after a meal. And we notice also the hunger reduction. The TT group were more satisfied than the AA group, which had the obesity risk variant. And then the ghrelin, obviously, the ghrelin reduction was much more along with the hunger reduction compared to the other group. So they have higher levels of ghrelin and feel hungrier even after their meals, and their brains respond differently to ghrelin and to the pictures of food, is what this last graph shows. So when we're working with individuals who are with obesity, it's important to factor in genetics because we're treating the whole person. Yes, environmental plays a role, but also taking into account that genetics is also playing a role. And that, yes, you can still have success, but it is part of the whole picture, the whole person. So these are additional resources. As promised, if you'd like to calculate your BMI, here is a trusted source. You could just add in your height and weight and you will have the information. Also, the impact of exercise on your DNA. And again, that's more of an environmental affecting your genetics. And then I do have an additional one if you'd like to read more about how FTO inhibitors affect obesity and cancer. And thank you very much. And if any of you have any questions, feel free to ask, and I'm happy to answer. And here are my references. And I hope all of you have a wonderful evening. Thank you.